Am I audible?
It is audible, Shruti. Start your presentation.
Yes. Good evening, everyone. My name is Shruti Gundale. Just sharing my webcam. There's some problem with webcam, I guess. Okay, I will continue. Good evening, everyone. My name is Shruti Gundale, and my research is based on morphoanalysis and phytochemical investigation of Albizia lebec bench seeds. This research was conducted by me during final year of my Bachelor of Pharmacy course. So Albizia lebec is a large erect tree which is found everywhere, and it is native to Indo-Malaya and northern Australia. So research have been done on various aspects of the tree, but seeds were not yet conducted. Research for seeds was not conducted. So Albizia lebec belongs to the family of Fabaceae, which is also termed as Mimosaceae or formerly known as Leguminosae. So my research consists of determination of ash values, that is which comes under morphoanalysis, and we also determine constituents present in Albizia lebec bench seeds by conducting various phytochemical tests. Also, we also performed atropine accuracy curve for determining the total alkaloid content in the seeds by using UV spectrophotometer. We did also conduct FTIR and NMR, but due to space constraints, I was not able to upload everything. So starting with the phytochemical test, the seeds were crushed by using mortar and pestle, and the extract from the seeds was obtained by using soxylate extractions for 24 hours. So various soxylate extraction was performed with water, methanol, ethanol, benzene, and chloroform. Each and every solvent was used for 24 hours to obtain extract. So for determining the phytoconstituents present in the seeds, for alkaloids, we performed Mayer's test, Wagner's, Dragendorf's, and Hager's test. For carbohydrates, Smolish test and Benedict's test. Same wise for glycosides, saponins, sterols, phenols, etc. We performed all the tests. From the results obtained from our test, we obtained that alkaloids were present abundantly in all the solvent system, that is water, methanol, ethanol, benzene, and chloroform. Carbohydrates were present only in water system, whereas they were absent in every other system. Glycosides were partly present in water, methanol, and ethanol, but not in benzene and chloroform. Seeds also possessed a high quantity of saponin, which we detected by using a solvent system of water. Steroids and triterpenoids were completely absent. Phenols were detected by using ferric chloride test, which we obtained in water but not in any solvents. Flavonoids were present in part system and part not. So in this way, we deduced which chemical constituents were present in Albizia lebec bench seeds. And moving forward, we determined ash value by the procedure given in IP, that is Indian Pharmacopoeia. So the total ash value obtained was 4%, acid insoluble ash was 0.5%, and loss on drying was obtained about 1.34. So when we determined that the alkaloid was present abundantly in every solvent system, we decided to measure total alkaloid content by using UV spectrophotometer. The spectrophotometer used was of Shimadzu company. So atropine accuracy curve determines the exact or not exact, means approximate value of alkaloid present in the seed. So initially, bromocresol green solution was used as an indicator against chloroform, which can give us the color change, which can be detected using UV spectrophotometer. Here we compared our results with atropine blank sample, and the results were obtained in the methanolic extract of the seeds. So the absorbance was obtained was of 0.3679, 0.3732, and 0.3698. Based on this absorbance, the linear regression equation was found out, which was found out to be y is equal to 0.0062x plus 0.0129, which is a standard error. Correlation coefficient was also found out, which was 0.9962. Correlation coefficient determines the extent of variation between the points we obtained on the graph. So this absorbance was then triplicated so as to obtain the approximate value. Total alkaloid content was found to be in the range of 57 and 58, which we found out to be the mean of 57.64 plus or minus 0.21, which we calculated as a standard error. So this was the base of my research. And in conclusion, I would like to say that Albizia lebec has been traditionally used for cure of various diseases. And the findings of our, including FTIR and NMR, may not determine the complete structure of phytoconstituents present in Albizia lebec bench seeds, but may sure help in deducing for structures further for scientists who may choose to research in this field. So various active phytoconstituents were found and and can be isolated using chromatographic techniques to further so that exact activity of these phytoconstituents in various diseases can be determined. Thank you.
Shruti, can you explain the selection of solvents? Various solvents used for the experiment is benzene, chloroform, ethanol, methanol.
Yes.
What is the order of solvents that is used for your extraction process and why?
Sir, initially we used water because many phytoconstituents are present in water. We wanted to use polar solvent as a water and non-polar solvent, which includes ethanol, methanol, and chloroform, and also semi-polar solvent, which is ethanol. So some of the phytoconstituents only get dissolved in that particular solvent. That it could be non-polar or semi-polar. For example, alkaloids, coumarins, and flavonoids are only detected in non-polar solvents. But not in, and flavonols are detected only in semi-polar solvent, but not in non-polar. So this was the basis of our solvent selection.
What is the polarity? Whether it is moved from polar to non-polar or non-polar to polar? What is the polarity changes?
The polarity change was from polar to non-polar because we wanted to determine whether or not the solutions, phytoconstituents are dissolved in which polarity are they dissolving, so that we can further separate it.
How you will determine the total alkaloid content?
Yes, total alkaloid content was determined by using UV spectrophotometer. We took the extract, then we treated it with chloroform and bromocresol green solution, and this solution was then observed in UV spectrophotometer against the blank solution of atropine, 0.1 micromolar solution of atropine.
Okay. And what is the formula involved in mean plus or minus SE? The tablet form you mentioned as a mean plus or minus SE. What it mean and how you will calculate?
Mean was calculated by the results we obtained in the by using the triplicate measures, and standard error was found out by using the standard formula of standard deviation divided by square root of number of samples we took. That was 3.
And the total ash value you got is 4%. What is the inference basing on that 4%? What you want to say?
No, actually it was just a parameter which we found out according to the IP.
Okay, how you will determine the acid insoluble ash? How you will determine the acid insoluble ash content?
Acid insoluble ash content, procedure?
Yeah.
Yes, so first we have grind all the seeds into a very fine powder, and then the total ash value, acid insoluble ash value, in which we have to first dilute that powder at 25 ml of HCl in 100 ml of beaker, and it was placed over wire gauze and boiled for 5 minutes. And it was then filtered with using ashless filter paper. And this filter paper was then ignited in the oven, and it was heat gently until vapor ceased to be evolved. And it was then pulled in the desiccator and weighed.
Okay, all the best, Shruti.
Thank you very much.